Hello everyone, this is Shinuja, Assistant Professor, Department of Biotechnology, St.Josephs College of Arts and Science for Women. Today topic is biological production of insulin. Insulin is a hormone produced by beta cells of islets of Langerhans of pancreas. It was discovered by Sir Edward Sharpe in 1916 while studying islets of Langerhans. The first licensed drug produced using recombinant DNA technology was human insulin, which was developed by GeneTech and licensed as well as marketed by Eli Lilly in 1982. Structure of human insulin. Chemically, human insulin is small, simple protein composed of 51 amino acid sequences and has a molecular weight of 5808 Dalton. Insulin hormone is a dimer of a A chain and a B chain which are linked together by a disulfide bond. Insulin consists of two polypeptide chain. Chain A, it is 21 amino acid long, and chain B, 30 amino acid long. Both chains are joined together by disulfide bond between two cysteine residues. Insulin produced by recombinant DNA technology. The basic step in recombinant DNA technology is similar for insulin production also. At first, suitable vector plasmid is isolated from E. coli and then it is cut open by restriction endonuclease. The gene of interest, that is insulin coding gene, is isolated from beta cells and inserted in open plasmid. Plasmid and gene of interest are recombined together by DNA ligase enzyme. Hakura et al. In 1977 chemically synthesized DNA sequence of insulin for two chains A and B and separately inserted into two PBR322 plasmid vector. These genes are inserted by the side of beta galactosidase gene of the plasmid. The recombinant plasmid were then separately transformed into E. coli host. The recombinant host produced proinsulin chain, that is fused beta galactosidase A chain and beta galactosidase B chain separately. These proinsulin chain A and B were separated from beta galactosidase by treatment with cyanogen bromide. The detachment of proinsulin chains from beta galactosidase is possible because an extra codon from methionine was added at N terminal of each gene for A and B chain. After detachment, A and B chain are joined in vitro to reconstitute the nabi-insulin by sulfonating the peptide chain with sodium disulfonate and sodium sulfate. This is the picture for human insulin production. Human insulin producing gene is taken from human pancreas cell and the plasmid is taken from bacterium. Then the human insulin producing gene is inserted in plasmid DNA. Then the recombinant DNA is introduced into the bacterium cell and then the recombinant bacterium is introduced into the fermentation tank and then extraction and purification of human insulin takes place. The need for preparing genetically engineered insulin. Insulin has been used for many years to treat diabetics. Diabetic is well managed by taking insulin. Earlier, insulin was extracted from the pancreas of killed cattle and pigs. It had shortcomings. It used to stimulate allergic reaction and other immune responses due to its foreign origin in some people. Another challenging was to cater to the ever increasing demand and large scale production. To overcome this, the production of insulin by recombinant DNA technology was done, and it is proved to be very beneficial. In fact, it was the first recombinant medicine to be used in the USA. Biosynthetic insulin produced by RDA technology is purer than animal insulin. It reduces the formation of antibodies against it. Advantages: large quantities of insulin can be produced at the same time. These forms of insulin is absorbed more rapidly than animal derived insulin, thus showing its effectiveness in a shorter duration. The insulin produced is relatively pure as purification techniques are performed after it is made, and it does not require the sacrifice of animals such as pigs and cows. It is cheaper than that isolated from pigs and cows. It does not cause any immune rejection like animal insulin. Recombinant insulin can be used for insulin therapy for diabetic mellitus patients.